Good afternoon, everyone. Hopefully you can hear me. My name is Richard Xiao. I'm the Director of Aging Research at King's College London. It's my pleasure to welcome you as the audience, but of course our speakers this afternoon for the webinar on biological aging and healthy longevity. But before we get to our speakers, I would like to say a few words about aging research at King's for those of you who are joining us for the first time. We are a consortium of researchers interested in healthy aging, longevity, using a life course approach from day one of conception through to the end of life and beyond. We're interested in the mechanisms of aging through to policy and also financial longevity. So a very broad consortium of researchers, both at King's College London and also with our global partners and industry collaborators as well. So welcome again to join us this afternoon. Of course, more information about aging research at King's is available on our website, which you probably would have seen in registering for this event. So over to our guests. It's my pleasure and honor to welcome Professor Terrie Moffitt and also her husband, Professor Avshalom Caspi. They are both our colleagues at King's College London, but for most of the year, they are based at Duke University in the Department of Psychology and Neuroscience, and also the Center for Genomic and Computational Biology at Duke. So it's an honor to welcome such distinguished academic guests to speak with us this afternoon. I won't go into the details about their biography because that's, again, available online on the event website, but I would like to highlight the really interesting work over the past decades to study biological aging with the Dunedin cohort, which they will be speaking about this afternoon, especially biomarkers of aging, DNA methylation, from mechanisms of aging approach through to the psychology of aging. And what I like about it is that this is across the life course. It's not only about aging when you reach a certain number. And we'll be hearing about their research and also perhaps some of their personal enjoyment of their research as well. And I noted in the biography says that Terrie likes to work on her poison ivy farm in North Carolina. I'm not sure whether you can be speaking about that, but that's certainly interesting. And also studying use of cannabis across the life course. I thought that was really exciting as well. So maybe we can have some discussion about that at the end. So without further ado, I will hand over to Terrie, and Avshalom will be speaking immediately after her. So thank you so much again for joining us, and we will follow up with our discussion afterwards. So if we can hand over to Terrie, if we can, there we go.
Thank you, Richard. I hope everyone can hear me there. I'm going to start the screen share here. Right, and I hope that that looks correct. Does it?
Perfect. That's wonderful. Good. Thank you.
Okay, so welcome everyone, and thank you very much for coming along today to Aging Research at King's. My name's Terrie Moffitt. For about the next 45 minutes, I'm going to Avshalom Caspi and I are going to tell you about our program of research on human development from birth to death. I apologize that I don't have a beautiful background like Richard does with a view of London, but at the moment we are here on the poison ivy farm in North Carolina. So I want to tell you a story about our work that's developing a measure of a human's pace of biological aging for use in prevention research. Now, you may know, those of you who know me, that I am not a gerontologist by training. Most of my career has involved the study of mental disorders, and mostly in children and adolescents and young adults. And this includes the last 25 years at the Institute of Psychiatry, Psychology and Neuroscience here at King's College London. So this story I'll tell you today is about a major discipline change for me late in my career. So this new work for our team started here. Now this slide shows you the well-known accumulation of multimorbid diseases across the lifespan that Richard referred to. And you can easily see that advancing age is the strongest risk factor for all non-communicable diseases. So if our aim as aging researchers is the prevention of the onset of these diseases, then the red arrow shows you that the ideal intervention point has got to be somewhere between the 2nd to the 5th decades of life. Now, I'm going to be telling you about quite a journey today, so I thought an outline would help us. And first, I'll start by telling you how our research works. Now, our team's been tracking the health and development of a birth cohort of 1,000 individuals born in 1972. It's the Dunedin Multidisciplinary Health and Development Study. The study members have by now reached their late 40s, so we believed that we were pretty well situated to contribute knowledge about the 2nd to 5th decades of life. In fact, the study is having its 50th anniversary this month. So now I'm going to introduce you to this study because it's our main research tool. Here you see the design. You see that at the top, there were 1,037 babies enrolled at birth in 1972-73. These were all the babies born in one city in one calendar year. There were no exclusionary criteria. Ethnically, this cohort is of white European descent. If you look down the left-hand column, it shows you the ages of data collection so far. There have been 13 assessments since babyhood, and each time we assess these cohort members, each of them spends a full day in our clinical setting for data collection. Down at the bottom in yellow, you see the last completed assessment wave was at age 45 in 2019 when 94% took part, and that was just before the pandemic began, and weren't we lucky? We didn't have an interruption. So because 94% took part, that means that the cohort still represents the full variation in the population. This is a cohort with no attrition bias or healthy volunteer bias. And down at the very bottom, you see we've been awarded funds to follow up the cohort at age 52. So we're very excited and we're planning for that now. Now, when we first proposed to study aging in young adults in their 30s and 40s, the peer reviewers of our grant applications at the MRC and the American National Institute on Aging scoffed at our proposals. Actually, they really hated our grant applications, and on the grounds that there simply could not be any variation on measurements relevant to the study of aging in people that young. This slide shows you composite faces from digital photographs of 10 female and 10 male Dunedin cohort members. And these photos were taken within 60 days of their 45th birthday. And they do look pretty young. However, these composites are 45-year-olds too. They comprise the 10 fastest aging in the birth cohort. So the first thing we observed was that there is definitely variation in aging among people many years before they're considered to be older adults. Now, all the first cohort were born in the same year. There's obviously enormous variation in our participants' aging, as you can see in their faces. So we set out to quantify this biological variation in order to study it. And to give you a bit of a background explanation of what we were trying to do with this research, I'm going to contrast three overly simplistic conceptual models. So on top is the model that I learned in gerontology courses that I took back in graduate school in the 1980s. I learned that disease onsets in late life, it leads to frailty and then to death. And we were told to get a disease and study that and study frailty. The middle bar shows a subsequent newer model, which gained traction soon after. It pointed to the importance of earliest life for aging research. For example, the theory of developmental origins of health and disease, DOHAD, pointed to perinatal factors that predicted late life disease and mortality. But these empirical findings about early life still left a black box in the middle of the life course. Now, it's truly remarkable that risk factors present at birth or in childhood can statistically predict late life disease, frailty, and mortality. But what we don't know is how this happens. So what's actually going on in the second, third, fourth, and fifth decades of life? The predominant assumption was that early risk must accelerate the aging process, and that's shown in the bottom yellow bar. But when we began this work, there was no empirical work testing this assumption of accelerated aging in the younger decades of life. So we decided that we could fill this gap. Now, when the cohort was first entering their 30s, we knew we wanted to grow this study of early child development into a study of aging. And so we began to collect measures that are used every day in geriatric medicine. I'm going to show you just a few photos of some of the assessments to give you an idea. We repeat these data collection protocols every time we assess the Dunedin study members as adults. These are just examples, and I'll go swiftly because these measures are already familiar to most of you. So first one you see here is postural hypotension. There's bone density and a DEXA scan. We have a suite of physical function tests. Here you see the chair stand. There is also an auditory assessment. Here you see detecting speech and background noise. There's a vision assessment session. Here's an optic nerve scan. And everybody's favorite is the dental examination. There's a respiratory assessment, and here you see whole body plethysmographic testing of lung function. There's a large number of clinical interviews that are interspersed between all of these medical examinations. So things like a life history calendar, psychiatric interviews, substance abuse assessment, financial knowledge, social life, and so forth. We draw a lot of blood for genomics and for biomarkers. And now when one aspect of the Dunedin study that makes it exceptionally valuable for the study of age-related change is that the study was designed to be multidisciplinary from the outset. Now, it's measured many of the constructs of interest to aging science repeatedly since the preschool ages. So the study by now has the longest running series of repeated clinical measures in areas such as hearing, vision, dental health, motor skills, and lung function, in addition to cognitive function, which I'm going to show you in a moment. Now, at age 45, we began brain scanning for the first time, and I'm showing you white matter hyperintensities here because, according to the neurology literature, white matter hyperintensities only begin to be detectable at about age 60. But the histogram on the right shows that we were able to find variation at age 45. And this is because of today's very sensitive scan resolution. You can now see these younger. Now, it's very sad that we don't have brain imaging data from earlier in life, but as you know, during the 1980s and 1990s, when the study members were young, scanning was simply not done on healthy children living out in the community. And in any case, in those years, in the 1980s and 90s, brain scanning was way too expensive then to scan 1,000 children. What we do have, though, is measures of the functions of the brain that were collected at age 3 in 1975. And here you see how each of the 1,037 children was individually tested as a 3-year-old. Now, from the data that were collected at age 3, we created a composite summary model to measure each child's brain function. Each child was examined by a pediatric neurologist. They took the Bailey motor skills test. They took the Raynell test of receptive language, the Peabody picture vocabulary test, and then after the testing session, when the child went home, the staff examiners rated each child's behavioral self-regulation, self-control. So an obvious place for us to start our work then was testing how key measures from gerontology research might be related to this measure of early life brain function. And now I'll show you some midlife outcomes. So cognitive processing speed. This is known to be a sensitive measure of age-related cognitive decline, and we've tested processing speed at every age, right up to age 45. Now, this slide shows you the relationship between age 3 brain function and age 45 processing speed. Across the bottom is age 3 brain function. On the left are the children with the least healthy brain function at age 3, and on the right are the children with the healthiest brains. 3-year-olds who had the poorest brain health now have processing speed almost a standard deviation lower as adults than their cohort peers who had the best brain health at age 3. We also measured the caliber of venules. These are microvessels in the retina of the eye. It's a non-invasive indicator of small vessel disease in the retina, but also in the brain. It's a widely used biomarker in research on risk for stroke and dementia in older adults. We found that 3-year-olds who had the poorest brain health had venular caliber almost half a standard deviation wider at age 45 than their cohort peers who had the best brain health at age 3. Walking speed is also a highly sensitive marker for age-related decline and for early mortality. It's a key measure in the study of functional aging. 3-year-olds who have the poorest brain function have gait speed almost a standard deviation slower at age 45 than their cohort peers who had the best brain function at age 3. So what does this mean? Well, when you begin a study of older adults and some of your participants show signs such as slow processing speed or wide retinal venules or slow gait speed or even poor episodic memory, it's not at all safe to assume that this reflects a decline of recent onset with etiology in late life. Individual differences on these key gerontology measures do have etiological roots very early in the life course. Now I'll tell you how we measured the pace of aging in the Dunedin study. Now, we thought of aging as a life course process, as Richard said, at Aging Research at King's. This is our mantra. So first, we know that exposures that cause age-related diseases accumulate starting from early life. We know that changes to physiology occur many years before diseases are diagnosed. We know that organ damage is difficult to reverse fully once it's established. And all of this together suggests that preventive interventions should begin early if we aim to slow aging and extend health span. We need more research to understand aging processes in young people to do this, because if we intend to intervene in these processes, we need to understand them. But how can we measure how fast a young person has been aging? The first thing we hit was a major barrier. To our surprise, there was actually no accepted measure of aging. Here's a 2020 paper, and it reports a lack of consensus. The authors are a list of some of the heaviest hitters in the field of gerontology. So it became clear to us that we were going to have to figure out how to measure aging. Now, I thought that our team ought to pay attention to what a group called geroscientists were saying about their operational definition of aging. They defined aging as the gradual, progressive, coordinated deterioration of physiological integrity across multiple bodily systems. So decline across organ systems that is synchronized and happening gradually and simultaneously. Now, in the Dunedin study, we had been tracking 19 biomarkers since the 1,000 study members were in their 20s, and you see these biomarkers here. They vary from periodontal biomarkers to cardiovascular, respiratory, HbA1c for prediabetes testing, 19 biomarkers assessed at age 26, 32, 38, and 45 years. Now, this slide depicts an eyeball look at the data for these 19 biomarkers. Virtually all of them did show gradual, progressive, coordinated worsening of physiological integrity, so they fit the geroscience definition. And this was across multiple bodily systems, again, fitting the definition. But we found that some cohort members have been declining fast, whereas others were hardly declining at all. So we wanted to capture these differences between individuals. We calculated 19 change curves, one for each of these repeated biomarker measures, and then we combined these curves to model what we called the pace of aging. Now, each individual in the birth cohort gets a score that represents their personal slope of gradual change that is consistent across all the 19 biomarkers. This captures that concept of synchronized longitudinal decline. I want to just explain this was vital to overcome a big criticism that research gets if you try to study aging in young people or midlife people. That is, if someone uses a cross-section of biomarkers to define biological age in young people or midlife people, how do you tell the difference between biological age and just being sick? So, for example, C-reactive protein could spike up because of a local infection, or lung capacity biomarkers could spike because of a temporary respiratory virus. But if you only use the extent to which many biomarkers across organ systems decline gradually together over years of time, then your model can ignore such short-term sickness spikes. The green histogram shows the resulting distribution of pace of aging scores in the Dunedin cohort. Now, most people, as you see there in the middle, aged 1 biological year per chronological year, as we should do. On the left, though, you can see there are some individuals who showed almost no biological aging between age 26 and 45. They've been remaining relatively young. And on the right, you see some cohort members aged almost 2 years per chronological year. They're on an accelerated aging track. We validated the pace of aging score by testing if it predicted cohort members' outcomes on all our measures of cognitive, perceptual, and sensory motor functional capacity. It did, and I'm not going to show you all of those findings. The full report was in Nature Aging just last year. What I will do is show you a couple of concrete examples. So here you see that the fast agers had more decline on tested cognitive function from childhood to age 45. This is comparing 2 IQ scores, one in childhood and one at age 45. Each data point on the slide represents 20 of the 938 cohort members who took part at age 45. The slow agers are over on the left. They have less cognitive decline. Inside the brains of the study members as well, a faster pace of aging score was associated with a thinner cortex and with smaller surface area of the brain assessed by MRI at age 45. Now, in this analysis, the brain is divided into 360 parcels, and the blue shows you the brain parcels that passed correction for multiple testing. So there you can get a feeling for what areas of the brain are most associated with rapid aging. Now, it's already well known what some of the best early predictors of early mortality are, and you see them here. There's familial longevity, Lowe's family social class origins, adverse childhood experiences, sometimes called ACEs, poor health as a child, low intelligence as a child, and low self-control as a child. Now, this slide shows you that all of these early predictors of mortality also predicted a faster pace of aging between age 26 and 45 as well. Now, just to summarize this section, the pace of biological aging can be measured in young people, and it validly predicts physical limitations, cognitive decline, sensory decline, and even facial aging and brain aging. But we needed an exportable version of the pace of aging that other research teams can use. You know, it's not very useful having a cool measure that you built from 20 years of repeated biomarker assessments if nobody else has those 20 years of biomarker data in midlife in their cohort. So, in fact, no other study does have those data. And that would mean that no other research team could try to replicate our work or to build on it and expand on it. And that would mean that our publications would never, ever be cited again, which would be sad to me personally, but it would be rather significantly disappointing to our dean at King's College London, too. So, with this in mind, we got motivated to develop an exportable version of the pace of aging. So now I'll tell you how we converted the pace of aging to a blood test called Dunedin Pace. Now, particularly urgent need for an exportable measure of the pace of aging is for testing the efficacy of anti-aging therapeutics. Now, in case you haven't noticed, we baby boomers do not want to get old and we do not want to die, and we're a huge and motivated market. We've got a lot of money, and products marketed as reversing our aging are all over the place now. So here's a great example, a daily capsule that promises to keep your telomeres from eroding at a very modest price. And for a little more light relief, last autumn in Iceland, I found this supplement that purportedly reverses aging. It's made from lobster shells, which is something they have a lot of in Iceland. Well, obviously you get the point that randomized clinical trials are needed. And quite seriously, this is a serious and growing biotechnology business. Now, as you know, there are government-funded clinical trials, there are compounds in the pipeline, and there are lots and lots of biotech companies who are working on therapies to fight aging. There are also many trials of behavior change interventions, such as physical activity or dietary management. Now, as a package, all of this work together is often referred to as the geroscience agenda. And the idea here is that advanced aging is a cause of all diseases. So if you can slow aging, you can prevent them all with one silver bullet. Now, these silver bullets are already available for yeast and worms and flies and mice. And as you know, research is pretty far along for treatments to slow human aging as well. The trick is it may work better to start these treatments as prevention before late life organ damage is too far along. Doing so could give them the best chance to show their efficacy. All these randomized clinical trials of prevention before late life that are being started up cannot use disease or death as their outcome variable. Those outcome measures are too far in the trial participants' future. You would have to run the trial for many years, which would be very costly. So randomized clinical trials of prevention for aging need a measure of the pace of aging. Such a measure could be useful for a pre-treatment baseline. You want to know who will respond best to anti-aging therapeutics, fast agers or slow agers. At this point, nobody knows. You could also use a pace of aging measure for tracking post-treatment outcomes. You want to use it for a long-term follow-up because we don't know yet whether treatment effects will last or whether they will fade out. This is simply unknown. So what is needed is cheap, easy, repeatable measures of the pace of biological aging to use as randomized clinical trial outcomes in prevention therapeutics. Now, a good measure has got to be sensitive to change in trials of anti-aging therapeutics. So I want you to remember that sensitivity to change is the goal that we're all working toward. So our next step was to translate our longitudinal pace of aging measure from the Dunedin cohort into something that could be done as a blood test in other samples and other studies. We did this translation by deriving a DNA methylation signature of the pace of aging. It's called Dunedin pace for pace of aging computed in the epigenome. We published the first version in eLife in 2020, and there's a new update this year in eLife that you see here. Now, this work required our team to learn to work with whole genome DNA methylation data, which our team has been doing for about the last 10 years. So this was, again, more new learning for me, a real career change. We used the longitudinal pace of aging measure that I've told you about as the training target, and then applied elastic net regression to identify probe sites in the whole genomic methylome of the Dunedin study members that would map onto variation between study members in their personal pace of aging score. The result is that researchers can now measure pace of aging in their own research participants, even if they don't have a longitudinal cohort study or, in fact, lots of biomarkers, but they need methylation data. Code to compute Dunedin pace is available on GitHub if you're interested in computing it yourself, and our team is standing by to help you with the technical work. Or you can also have it done commercially by a nice company called True Diagnostic. Now I'll talk about how Dunedin pace differs from the clocks. Now, there are several epigenetic age clocks already developed that you will know about, beginning with the Horvath clock, the Hannum clock, Grimm age clock, Pheno age clock. You'll be wondering how Dunedin pace is different from these. Now, pace differs from the clocks in a number of different ways that are fundamental to its validity. The timing of this talk, unfortunately, doesn't allow for me to explain all of them, and they're a bit technical, but there is one point that's relevant for using a measure in a clinical trial to test an anti-aging therapy. The clocks assess how much aging has occurred since conception, but they do not pin down when in life a person had accelerated aging. Was it recently, or was it aging accelerated as a fetus, or during childhood? We can't tell from the clocks. Contrast, Dunedin pace was trained on biological change, and it's designed to measure how fast a person is aging now. So we don't call it a clock. It's more like actually a speedometer. We're now publishing validity checks outside the Dunedin cohort in other research studies, and I can swiftly show you just a few of the key ones that people usually look for in a good measure of aging. Now, this slide shows that Dunedin pace shows the expected black-white difference in the pace of aging, and these are data from the American Health and Retirement Study, panel of several thousand Americans. Down at a pace of aging is shown in the very bottom panel. African American older adults are aging faster than white Americans. You also might notice that the Dunedin pace gives the same effect size for ethnic differences as the Grimm age clock. This happens a lot when they're compared head to head. They both tend to perform well, but for different reasons having to do with how they were designed and derived. Here, Dunedin pace predicts mortality in the Framingham Heart Study. The red line shows you that there's earlier death for people who scored as faster agers on the Dunedin pace measure. This slide compares DNA methylation aging measures in ADNI, which is the Alzheimer's Disease Neuroimaging Initiative study. The clocks are on the left and Dunedin pace is on the right. PACE so far has the best capacity to differentiate among groups who have no cognitive impairment, they're in blue, mild cognitive impairment, shown in yellow, or people who were diagnosed with dementia in the ADNI study, shown in gray. As you can see, the dementia patients have been aging the fastest. replicated this finding for dementia in the Framingham Heart Study offspring cohort. This is a longitudinal analysis that included 2,264 participants with 14 years of follow-up time for dementia ascertainment after the methylome was assayed. The figure shows survival curves for 3 groups of participants: those with Dunedin pace 1 standard deviation or more slower than the mean, that's the blue line. Those with Dunedin pace within 1 standard deviation of the mean or average aging, that's the purple line, and those with Dunedin pace at 1 standard deviation or more faster than the mean for Framingham, that's the red line. And what you can see is that participants with faster Dunedin pace at baseline were at increased risk of developing dementia over the follow-up period at an average age of 82 years old. This is potentially important because funders are intending to, government funders are intending to fund midlife prevention trials for dementia. But for these prevention trials, an outcome measure is needed that can be used in people under 60, because it will take too long to wait until midlife trial participants under 60 reach their 80s to see if they get dementia or not. So this could be an initial way to assess how well an intervention is doing. The next validation check I'll show is from Calorie. That's a randomized clinical trial of caloric restriction. This trial addresses the question of whether Dunedin pace is sensitive to change from anti-aging therapeutic treatments. Now, miraculously, the Calorie team convinced people to be randomly assigned to a treatment arm, and they had to strive to eat only 1,800 calories a day for a whole year, something that I personally could never do. But they did it, and here are the calorie data. The gray line shows the effect size for the difference between the treatment group and the control group, as tracked from baseline to 12 months to 24 months, that is post-treatment. The first and second generation clocks are shown on the left. They did not capture the treatment effect as clearly as Dunedin pace, which is the one on the right. Now, the trial arm who restricted their caloric intake slowed their aging as compared to the control group who ate as normal. And the control group kept on aging 1 biological year per chronological year, as they should have. Now, this, I caution, is a very small effect size, is 0.25 standard deviations. But it's early days in using DNA methylation measures in randomized clinical trials, and this is one of the first findings that we have. Now, in the Texas Twin study, Dunedin pace is accelerated in adolescents who experienced early life adversity. Family disadvantage is shown on the left in pink, and neighborhood disadvantage is shown in the right, on the right in yellow. So Dunedin pace is sensitive to early life adversity. And with our colleagues at Exeter University, we exported Dunedin pace to 7 case control studies that have been comparing people with schizophrenia against matched controls. Now, it's already known in the psychiatric literature that patients with schizophrenia tend to die on average 10 years early. And here what you see is a meta-analysis that we did with our Exeter colleagues. It totals about 5,000 individuals in these studies, and it shows that Dunedin pace was faster in people who have schizophrenia than in matched controls in all 7 of the individual studies. The red diamond at the bottom shows that there was an overall moderate effect size of 0.6. Now, you may recall that I began by saying that I am not a gerontologist, I'm a clinical psychologist. I'm motivated then to know how we can use the pace of aging to answer some practical questions that will really help people. And one idea that drives my team is the idea that mental disorders are a neglected prevention opportunity to improve aging. So at this point in the talk, I'm going to hand over to Avshalom Caspi. Avshalom is my collaborator, but we're also married to each other. And he also is a professor at King's College London in the Institute of Psychology, Psychiatry, and Neuroscience, and also at Duke University. So, Avshalom, would you like to join us?
Yes. We're sharing the same screen. Thank you, Terry. Pleasure to join you. What I would like to do next is show you evidence that early life mental disorder history is important for successful population aging policy. I'll show you that people with mental disorders will age younger and faster, that they'll have more chronic physical diseases as they age, and that they're also more likely to develop dementia and to develop it earlier. This population pyramid shows the prevalence of mental disorders and that it peaks between ages 10 to 40. Now, keep in mind that the onset and prevalence of physical diseases peak after age 50. In contrast, mental disorders have a similar prevalence to physical diseases, but they peak earlier in life. So this time scale sets up the hypothesis that successfully treating people with mental health problems in early life might actually prevent age-related diseases later in life, because we know that mental disorders are in fact treatable. In 2019, we published this viewpoint essay in JAMA Psychiatry. Here we put forward the hypothesis that the people who have early onset mental disorders in the first half of the life course will tend to be the same people who have multi-morbid physical diseases in the last half of the life course. If these are in fact the same people, then mental health care might have a great opportunity to prevent the rising burden of age-related diseases. And so we set out to test if in fact these are the same people. And first we turned to the Dunedin study. So how did we measure mental disorders in the study? In Dunedin, we've used here our 5 decades of repeated mental health assessments to build a composite measure to summarize each person's mental health history. We've assessed 17 different disorders, and people who had high scores on this composite measure had younger age of onset of their first mental disorder. They had a greater number of different diagnoses that accumulated over their lives, and they also had longer duration of years lived with a disorder. So that's how we summarize the entire life course history of mental disorder in the cohort, beginning in adolescence. Now, this slide here gives a vivid illustration of the connection between mental health history and rapid aging. The 1,000 Dunedin study members are divided on this slide for presentational purposes into 6 groups based on their level of mental health history. Those with the least mental health history, never having really any symptoms of mental disorder, are on the left. And those with the greatest history, the most extensive history of mental disorder, are on the right. They had early onset, they had more different disorders, and they lived more years with disorder. And what you see is the group of study members with the least versus the greatest psychiatric histories are 3/4 of a standard deviation apart on ratings of the age of their faces. This represents a large effect size. So this is just to document, as it were, that there's face validity to this idea of mental disorders being associated with accelerated aging. But to achieve more precision than simply looking at facial photographs, we tested the longitudinal measure of the pace of biological aging that Terry described to you. Recall that we measured the biological pace of aging from ages 26 to 45 using 19 different biomarkers. And what we found is that people with the most extensive history of mental disorders are aging about 3 months faster per year on average. So an important point here is that the fast aging people with a psychiatric history remained the link between psychiatric history and biological aging remains significant after we controlled for two behaviors that are known to accelerate aging, namely tobacco smoking and also for the use of anti-psychotic medication. I'd like to do now is leave the Dunedin cohort for a moment. And for this last project, what I'd like to do is I'd like to tell you about how we've accessed administrative registers of medical records for the whole population of New Zealand. So we're now moving from a sample in New Zealand to studying the entire population. And here we wanted to test our hypothesis that people who have mental disorders, right, when they are young, and people who have physical diseases when they are old or older are the same people. And we wanted to test this again at the level of the population and over a wide age range. Here we defined our study population in the New Zealand National Registers as those who were born between 1928 and 1978. This is about 2.5 million people. We obtained medical records about these individuals from 1988 to 2018 over a 30-year period. And importantly, each encounter with the medical system is here dated. And so we also have information about billing information and stays and lengths in hospitals, and I'll show you a bit more about that. And the age period that we've assessed here covers a 70-year span between essentially 20 to 90 years. We designated the first admission for mental health treatment for any of these disorders as our index event. Substance use disorders, psychotic disorders, mood disorders, and so forth. Then we tested for subsequent later onset of any of these physical diseases, heart disease, gout, COPD, diabetes, and so forth. It's important to note that we were also able to control statistically for cases where a person had been treated for a physical illness at a date before their first mental disorder onset, and for time also outside the country when an illness would not show up in the New Zealand medical records. Here is a representation of the results. The purple bars show you the prevalence of subsequent physical diseases for people who had a prior mental disorder. The blue bars show the population-based rate of chronic physical diseases for people who did not have a mental disorder history. And what you can see, and the blue bars again are for people with no mental disorder history, the purple bars for people with. What you can see is that every chronic physical disease that we studied was elevated for people who had a mental disorder earlier in the past 30 years. Whether we look at gout, whether we look at lung disease, whether we look at heart disease, whether we look at cancer. Now, we know, in fact, from other studies that people, for example, who are diagnosed with schizophrenia are more likely to develop later physical diseases. But what is really interesting and I think important about these series of findings is that we found that all mental disorders carried about the same elevated risk. What you can see at the top is the link between any mental disorder, right, and subsequent physical diseases. And then below that, in the black dots, what you see are the associations between specific mental disorders and later physical disease. Again, they're the same, whether we look at substance use disorders, psychotic disorders, mood disorders, and so forth. More recently, we've also tested our hypothesis more specifically for the outcome of Alzheimer's disease and related dementias. Here we found that the prevalence of Alzheimer's dementia was 4 times higher in people who had a mental disorder in the past 30 years. This analysis is restricted to 1.7 million individuals in New Zealand over the age of 50. This report was published just 2 months ago. This is here a result that we found especially interesting, namely that in the New Zealand population disorder data, mental disorders predicted dementia better than did chronic physical diseases. So mental disorder histories here are shown in orange, physical diseases history are shown in purple, and this is especially true for people born in the 1950s and 60s, and I think there are a few explanations for this historical difference, and we can speculate about that perhaps later. Finally, we've also looked at mortality. The important thing here is, in this particular analysis, is that people with mental disorders earlier in life, in orange, died young just as often as people with physical diseases did later in life. Mental disorders are just as important for early mortality as are physical diseases. But mental disorders also occur much younger, giving us an opportunity for preventive treatment. This slide shows you a report from the American National Academy of Sciences seeking to identify the primary social determinants of early mortality. In 2021, this was the most downloaded report from the National Academies. And yet, mental health is not mentioned anywhere in it. Mental health really is a neglected prevention lever. This is a photo from a trip to Thailand a few years back, and we decided to share it with you because I think it carries just the right message. If you don't take anything away from this talk, I hope that you'll take away the message that the opportunity to improve the mental health of a young person is really a very special gift. Done right, it might improve their health span, and it might also bring a large return on investment for decades into the future. We need to find out if successful treatments for mental disorders can actually slow aging and also prevent chronic physical diseases. We could start, for example, by measuring biological aging in randomized clinical trials of treatment for mental disorders. We could also follow up the medical records of people who took part in successful psychiatric clinical trials to see if they had fewer diseases years on. So these are ideas for future research. I'd like to thank our colleagues and research teams at King's, at the University of Otago in New Zealand, and at Duke. And finally, I'd like to thank ARK for inviting us to join this afternoon.
Thank you very much, Avshalom, and of course, Terrie. This was a wonderful tour de force and very insightful. Lots of questions. Although we only have 5 or 6 minutes until the end of this webinar, I'm sure the audience would be able to email you directly with their questions as well. Now, we do have a few questions that have appeared in the Q&A, and I'd like to invite the attendees to post further questions. If we don't get through them, I will forward them on to both Avshalom and Terrie. But first of all, I would like to ask a few of my own questions. And of course, this is well aligned with everything that we're doing at ARK and across King's College London. And one important point that Terrie mentioned is the use of worms and mice and flies and so on that account for the majority of academic aging research at the moment. We really do need more cohort studies and humans because I feel, the animal models, the experimental models, do not resemble the stresses of human life. They don't have bank accounts or wallets, that actually is what we are thinking about most of the time. And they add on to our stresses. They don't breathe the air that is polluted and the water, and they don't have, certain foods that we all love to take. So we do need to translate much of the basic medical research into cohorts like Dunedin and others. So that was a very important point. And for Avshalom, you really highlight the importance of mental health and stress. And one thing I was thinking about is the impact of, for example, the pandemic. Many are suffering from PTSD through many reasons, not only because they had the symptoms of COVID, but because they lived through a very difficult time. And what impact that has on biological aging. I think that's really important, not only mental health that everyone's talking about. And a second example is, unfortunately, the war in Ukraine. There are so many displaced children and young people who will be, impacted throughout the rest of their lives, just based on the stresses of the situation. Do you have any comments on those?
I will say that one of the interesting features of the pandemic is that for those researchers who will be following up individuals who sort of lived through the pandemic, a real challenge will be to separate out the effects of the pandemic for those individuals who were ill from the effects of the pandemic that are registered via the upheaval and the stress that it has generated in life. So that's going to be a really interesting, I think, research challenge to separate out those two routes by which the pandemic may have long-term effects.
What you said, Richard, about the humans' lives being different from the laboratory animals like worms and fruit flies is true. But in case watchers have not seen this, there are studies on the west coast of the United States in Seattle, Washington, going on of aging in pet dogs who live at home with humans. Now, these pet dogs don't have a debit card or a wallet, but they do breathe the same air and experience many of the aspects of life of their human owners. And so Daniel Promislow and Matt Kaberline are the investigators, and that's a good study to watch of pet dogs aging.
That's great. Thank you for reminding us about the dog study. I know Matt very well, and I think that's a great study as well. And even to look at the pace of aging in us, whether we have pets or we don't have pets. I think that's an important aspect, at least for stress or anti-stress in and having pets. Now, one area which I'm also interested in is, as a vascular biologist, is using the retinal scan. I found that fascinating, and how we can have a window to the brain through the eye. I think that's an important aspect, that we can better understand the importance of perfusion, oxygenation of the brain, and vascular function, and how we can translate that information in through to cognitive function. I think it requires a lot of research over the next decade, both at King's and globally. So I think, thank you for highlighting that. Maybe another quick question. Yeah, go ahead.
I may just add, the group at Singapore, with whom we've been very fortunate to work at the Singapore Eye Research Institute, have also come up with an automated way of scoring retinal images, which bypasses much more of the laborious work of grading each scan. And if anyone is interested in that, I'd be happy to share the paper that appeared in Nature bioengineering. So that's a new tool that is available and can be used by many research groups.
Yeah, Tian Wang is the leader of that group,
Right.
and this new technique substantially reduces the cost and time of doing the retina research.
Great, thank you for that information. There are two more questions which I'll briefly mention in the Q&A. The first one is, many thanks for a great talk. It would be interesting to include pace of aging in clinical trials. What would be recommended the minimum period for sensibly measuring this in sample sizes for phase 2 trials? Weeks, months, or years? So any comment on that? That was a question for Mittel.
That's really the holy grail. That's what we're all trying to figure out at the moment. One approach to that is to study cohorts studies that have multiple waves of methylation data, so that you can look if there has been change in the pace of aging among participants who say quit smoking between sweeps or lost weight. But as many clinical trials researchers have explained to us, if you want health behavior change, you need to have rapid change in your outcome variables, because participants want to see that their health is improving within weeks or months, not years. So we don't know how fast these clocks or the pace of aging might change. And that's one of the things that we're going to be working on next. It's very high priority.
Great, thank you for that question, Mittel. Another question is whether you know if medications taken for mental health contribute to premature aging in association with mental health. And from that perspective, I know at the beginning I mentioned about cannabis. What does that do to the pace of aging? Does it help? Does it increase? Fascinating area.
This is a really difficult question to sort out. What I can tell you is give some empirical sort of information here, which is that in the Dunedin study, for instance, we have been able to rule out the effect of, say, antipsychotic medication on the link between mental disorder history and accelerated aging. In the analysis that Terrie showed you of the link between schizophrenia and pace of aging measured via DNA methylation, we've also been able to show that individuals who are on antipsychotic medication are not aging any faster than those with a similar diagnosis who are not on medication. So that's, it appears to be that the association is not entirely attributable to antipsychotic medication. Now, with regard to other substances, again, with schizophrenia, it's a real challenge because the rate of tobacco smoking, for instance, among individuals diagnosed with schizophrenia is so high. So it is imperative that we rule out the association with those substances. What I can say about that is that the associations are attenuated quite a bit when we control for substance use history. They remain significant, but that also the misuse of substances is in fact part and parcel of the very condition. So we can partition variants all we want, but we often do so at the risk of actually no longer studying the very condition that we need to be treating. But this is a really important
Do you want to
issue.
say what the finding is about the pace of aging in cannabis?
So this is new work in progress. Thank you. This is why we work well together. Right?
I can even nag him in the middle of a talk.
So we do actually. This is a manuscript that isn't nearly complete. There is in fact a link between long-term use of cannabis and the accelerated aging. Most of that is attributable, however, not to cannabis per se, but rather to polysubstance use. That is that persistent users of cannabis, individuals who have been using cannabis for a long time throughout their lives, also use many other substances, tobacco, alcohol, other illicit drugs. And what really makes up the link between cannabis users between cannabis use and accelerated aging is, in fact, that profile of other drugs.
Great. Well, thank you very much for that discussion. I'm really sorry that we've run over time slightly. As with all of these discussions, 1 hour is never enough, but we do look forward to welcoming you back again many times in the future, especially when you are in London. We hope that we can see you in person. So with that, I would, yeah, I know. So with that, I would really like to thank you both again for a wonderful talk, wonderful discussion. I could spend another 1 hour if you have time. We can remain online and discuss some of these issues. But I'd also like to thank all of the attendees for joining us this afternoon, or wherever you are, whatever time zone you're in. And of course, the talks are recorded and will be made available online on the event webpage, so that you can go back and view it again. So with that, thank you so much again to all of you for joining us. Terrie, Avshalom, it's been a pleasure hosting you this afternoon, and we look forward to working together over the next few years. And see you next month in London.
See you soon.
Thank you.
Thank you so much, everyone.